Hi guys, welcome back to another biological topic discussed by me, Josh W. Today we're discussing the protein folding process. So the starting point, proteins are built of amino acids. They can also be broken down and refolded. This process of the protein being unfolded is called denaturation. And this denatured or unfolded protein is our starting point. This is what you need to keep in mind because this is where we're starting for this process, okay? So the first general step in protein folding is the hydrophobic collapse. This is the first step needed to get a denatured protein to a folded protein. This is the coming together of hydrophobic amino acids from far apart in the sequence. And this happens because of the hydrophobic effect, which pushes amino acids that are nonpolar away from the water. And here we have a GIF that's showing this. As you can see, all the hydrophobic amino acids being pulled together. Now back to the slides. So the weak bonds hold the forming structures together. And as you can see, or you might be able to see, but behind me of James Bond, it's not that bond. But there are three main types of weak bonds. We have the electrostatic interactions, which are ionic bonds or salt bridges. We have hydrogen bonds, which occur when hydrogen is covalently bonded to an electronegative atom. These are the most common. And we have our van der Waals interactions, which happen with molecules with no charge nor polarity. Also, there are disulfide bonds, which are a type of covalent bond that occur in between two sulfur atoms. Interactions can either be long range or locally. Long and local range interactions can take place at the same time, and this causes the formation of the native state. Remember, the native state is important. As discussed earlier, the hydrophobic effect draws together hydrophobic amino acids from far apart. The long range, this is the tertiary structure. Local interactions pertain to secondary structure formation through hydrogen bonding or close together. We'll now dive deeper into the different structures formed. Two of them were mentioned above. So the protein structures can be categorized into four main groups. We have our primary structures, which are amino acids in a chain sequence. We have our secondary structures, which are hydrogen bonds with peptides. We have our tertiary structures, which are 3D folding pattern due to side chain interactions. And our quaternary structures, these are proteins consisting of one or more whole amino acid chains. So think two amino acids being added together, that's a quaternary structure. And these structures are formed due to different actions and interactions during the folding and subsequently the unfolding and repetition of this process. So we have different protein shapes. Of course, we have our primary, our tertiary, our secondary, our tertiary, and our quaternary. As you can see, there are multiple. Not all folds are folded equally. No, but seriously, some folds are incorrect and thus must be folded and tried again. These incorrectly folded proteins are known as misfolded. Chaperone proteins exist and are able to help to protect the proper folds and the hydrophobic residues. Intermediates also have a huge role in defining how a protein folds and its pathways. Also, the slightest discrepancies can lead to misfolding and aggregation. Aggregation or clumping of proteins as well as misfolded proteins are non-functional and can also lead to diseases. So always looking. So as you can see, we have our native state and then we have our denatured state. This is a popular meme with younger generations. This is the native state and it's always looking for the lowest energy level. And here we have our pissed off denatured state because it wants the lowest energy level. These are the resources used to produce this video. Thank you for learning about protein folding with me today.